Hello, everyone, and thank you for joining my presentation today. I'm going to talk to you today, and I hope by the time that this presentation is over, you'll have a better understanding of immunology and how that relates to swine production and health. So for that, we're going to go through this outline, this presentation outline. We're going to start with just an introduction to immunology. Start with the innate and the adaptive immunity and the difference between the two. Talk a little bit about immune cells and their functions, and then what happens when the immune system encounters antigen or a pathogenic population of bacteria or viruses. What's the response to that pathogenic challenge in the animal? I'm going to spend a little bit of time talking about major histocompatibility complex molecule. We don't hear a lot about that, but really it's key for the whole immune response to pathogenic challenges related to antigen presentation. And so we're going to spend a little bit of time explaining that, and hopefully you'll have a better understanding of how that whole immune system integrates itself to combat a pathogen. Then we're going to talk about inflammation and regulation of the immune response and how that relates to livestock production or pig health and productivity. And then at the end of the presentation, I'm going to go through a set of data that will explain how we look at immunology when we're measuring the actual immune responses in the pig, and then how to interpret that as it relates to the pig health and productivity. So to start off with, the immune system is divided into two broad kind of categories or branches. The first is the innate immune response, and then adaptive immunity. And in these circles here, this first larger circle is showing all of the cells that make up the innate immune response, or the cells of innate immunity. And then the smaller circle here shows those cells that make up the adaptive immune response, or adaptive immunity. And these two in the middle, the gamma delta T cells and the natural killer cells, have aspects of both the innate immune response as well as adaptive. We're not going to focus on those for this presentation. We're mainly going to focus again on cells within the innate immune response and cells within the adaptive immune response. And the biggest difference is specificity to the antigen or the challenge that the immune system is responding to. For the innate immune system, it's non-specific, meaning if it detects anything that's not the host or not self or any pathogenic challenge, it responds to that, kills the bacteria or the pathogen, and communicates to the adaptive immune system. The adaptive immune system is made up of B cells and T cells, and it's your antibody response and your T cell response and your memory cell response. So whereas the innate immune system is very quick and fast acting, the adaptive immune system actually takes a little bit longer for it to mount that immune response to eliminate a pathogen. And we're going to talk in a little bit more detail about how these two systems of the immune system interact. And then before we go on, I do want to mention, oftentimes it's not thought about, but the epithelial cells of the intestinal tract or even your skin actually act as that first immune response of the body. So your skin cells protect the bacteria from being able to get into your systemic circulation, just like the intestinal epithelial cells are the first line of defense of the innate immune system, preventing bacteria from getting into the host. And then before we move on, immunology is a little bit like learning a foreign language. As immunologists, most of what we learn in immunology really is the language of immunology. So some common terms that you'll encounter when you're talking about immunology or hearing someone speak about immunology, the first is leukocytes. And leukocytes are the same thing as white blood cells. So those are the immune cells of the body. All of the immune cells of the body are white blood cells, and they're all leukocytes. They're the same thing. Lymphocytes refers only to B and T cells. So when you hear the term lymphocyte, it's only referring to those cells of the adaptive immune response. So, and one way to think about this, you'll hear these terms used interchangeably, but that's actually incorrect. So all lymphocytes, the B's and T cells, are also leukocytes, but not all leukocytes are lymphocytes. So there is a distinction between those two terms, and it really does matter they really can't be used interchangeably if you truly do mean the lymphocytes of the adaptive immune response. So moving on to talk about the innate immune response in a little bit more detail, and particularly this barrier function along the gastrointestinal tract. Because again, I don't think when we think about immune system responses, we don't really think about the epithelial cell layer as the first line of immune defense and that barrier function in the intestine. So when you're in the gastrointestinal tract, there are actually kind of three layers of the innate immune response, or the innate immune barrier to protect the host. The first is the microbial barrier, which is made up actually of the commensal bacteria along the gastrointestinal tract itself. So those organisms that are present but are not disease causing. And that's in this outer layer of mucus along the lining of the gastrointestinal tract. One layer deeper of this immune barrier function is the chemical barrier. And this is the thick mucus layer that protects the epithelial cells then from the lumen of the gastrointestinal tract, for example. And within this chemical barrier, or this mucus barrier, there's the mucus itself, as well as certain antimicrobial peptides that the epithelial cells themselves may produce to protect them from invading bacteria. And then the physical barrier itself, which are the epithelial cells that line the gastrointestinal tract. And really, that's kind of that first line of defense, and is considered part of the innate immune response. And then to talk a little bit about the big picture of the immune system and the immune response. I listened to a lecture given by Dr. Gary Huffnagel, and he's an immunologist that focuses on the immune effects of probiotic bacteria in human health. And when I heard his presentation, he actually made a statement that I thought was pretty radical at the time. He said, the immune system's role is not to combat against disease or to eliminate or control diseases and disease challenges in the host. And I said, wait a minute, what do you mean? It has to be. That's exactly what the immune system does. It combats pathogenic challenges to the host. But if you think about it in a little more detail, it starts to make sense. Because he said that if the immune system's role was to combat disease or fight pathogenic bacteria, it's doing a horrible job. And if you think about, again, this normal microbiota that's along the barrier of the gastrointestinal tract along the epithelial cell layer, he's right. There's many bacteria that are considered non-self or not the host, some of which have the potential to be very pathogenic. But when everything is in a normal state, or homeostasis, a steady state, the immune system samples those pathogens, those potential pathogens. It responds in a way that says, oh, okay, you're there, we know you're there, but everything's fine. It's only when there's a sense of dysbiosis at the level of that gastrointestinal tract, shown down here at the bottom, where there's a disruption in that innate immune barrier. Those foreign pathogenic challenge bacteria can then get through into the host, into the system of the host, that the immune system starts to mount its defense. So really, the immune system's not necessarily combating pathogenic bacteria or pathogenic organisms. It's really trying to bring that host back to homeostasis or to a steady state. And how does it do that? How does the immune system go about bringing the host back to a steady state? Well, it does that by fighting disease. So it can distinguish the bacteria are foreign entities, foreign pathogens, and it'll go about eliminating those pathogens when homeostasis is disrupted. The fever response is really just a response to the immune system's response to eliminate that pathogen. And then that resolution of fever is an indication of a regulation of the immune response. So again, that whole fever response and then resolution of that is to bring that host back to a homeostasis or steady state. The chemical controls themselves, the hormones and cytokines, particularly the cytokines produced by the immune cells, regulate these body activities to bring the host to homeostasis or to maintain homeostasis in the host. An example of this is when you get sick and there's an immune system response, they produce cytokines that act as hormones that decrease your feed intake response. And our pigs are the feed intake response. So that's why when you're sick, you don't want to eat. And when your pigs are sick, they tend to go off feed. So all of those mechanisms are ways to bring that host back to resolve homeostasis or steady state. And then often not thought about as an immune response, but the process of healing damaged tissue is really an immune response to bring about host homeostasis. So the example, if you cut your skin, the immune system responds to that to eliminate any potential pathogenic bacteria that may get in there and infect that cut. And at the same time, it starts to eliminate or clean up those damaged tissue cells or dead cells that are there as a part of that injury. And that whole healing process of eliminating the damaged tissue, combating any foreign entities that may be trying to infiltrate the host through the cut, as well as generation of new and healthy tissue, is all part of the immune response trying to maintain homeostasis. So again, if you think about the immune system from that aspect and not just combating or fighting off foreign pathogens or disease of the host, the immune system works as a holistic operation to really just, again, maintain or bring back homeostasis to the host. And if you start to think about that, it starts to make a little bit more sense when you start to see immune factors that are measured in our host animals related to health and performance. So the main players of the immune cell, before we start talking about the process, we're going to talk about some of the main players that are keys to an immune response to a foreign pathogenic challenge. And the first one is the neutrophils. And these are innate immune cells. They're the first line of defense. They're the first cells that are going to be going into that cut. When you get that cut on your skin, they're the first cells that arrive there. And they're called to the site of infection by other immune cells. And I've given you a picture of the cells, each one of the ones we're going to talk about, a picture of it under the microscope. And neutrophils are called multinucleated because they look like they have, in this picture, 3 different nuclei that are present within that cell. But really, the nucleus of the neutrophil, they really only have one nucleus. It just twists around. But it's a very nice identifying characteristic under a microscope when you're looking at these cells under a microscope. The next immune cell is the macrophage or the monocyte. It's a monocyte when it's in blood, and it becomes a macrophage when it leaves the circulation and goes into the tissue. And these are innate immune cells as well. And they are what we call phagocytic, and they are antigen presenting cells. And we're going to talk about this in a little more detail in some of the slides to come. But again, to identify the macrophage, the monocyte in the blood is shown here on the left. It has a kidney bean shaped nucleus with a fair amount of cytoplasm around that cell in the lighter purple. But when the macrophage goes into the tissue, when the monocyte moves into the tissue and becomes a macrophage, it still has that kidney bean shaped nucleus, but it now starts to have appendages and vacuoles within the cytoplasm. And we'll talk about the difference in that when the macrophage is kind of in tissues and its activity. And then the last 2 cells that we're going to focus on today in this presentation are the B lymphocytes and the T lymphocytes. And B lymphocytes are cells of the adaptive immune response, and they're the ones people are probably most familiar with because they're the antibody producing cells. And they also have the capability to produce antigen, but really what they're known for is antibody machine factories. They just produce antibodies in response to immune challenges. And then the T lymphocytes are also cells of the adaptive immune response. And they're categorized in 2 categories with 2 very different functions. The T helper cells coordinate the immune response. So they produce a lot of cytokines, those communication molecules that orchestrate really the whole immune response to eliminate a pathogen. Whereas the cytotoxic T cells are a little bit of a different flavor of T lymphocyte, where they actually kill damaged or infected cells. In the case of, let's say, a virus infects a host cell, the cytotoxic T cell detects that virus is in the cell and then eliminates the cell so the virus can't replicate and cause its viral disease. Now, if you look at these 2 under the microscope, you actually can't distinguish a B cell from a T cell just using the microscope. They both look the same. They're largely nucleated. The nucleus is very, very large in the T and the B cells, with a tiny little ring of cytoplasm around the nucleus in those cells. And, you know, they do a lot of cell division. So therefore the nucleus of these cells is more robust, say, than some of the cells of the innate immune response, because they do cellular replication in response to an immune challenge. The only way to tell them apart really is to look at cell surface markers on their cell surface. So they do have distinguishing markers that will distinguish between B and T cells. And we're not going to talk a lot about that today, but you can use flow cytometry to determine that, the difference between those two lymphocytes. But you can't tell the difference under a microscope. So now that we kind of got the main players, we've learned the names of the main players and their functions. What happens when these cells encounter antigen? Or an antigen is really defined as a challenge, some foreign entity, or some entity that's not self or not the host. So what's that whole process look like? So the first response that happens, of course, happens from the innate immune response. And in this schematic, we have a macrophage here in tissue, and it detects the bacteria that are invading the host, here represented by the little yellow dots. And when it detects that on its cell surface, seen here with the macrophage detecting that bacteria, it starts to produce a series of cytokine molecules or cytokines that act really as communication signals to the rest of the immune response. And you can think of these cytokines as essentially smoke signals that the macrophage sends out that communicates with other immune cells. And one of the cells that it communicates with and kind of leaves a trail for how to get to the tissue, it communicates with this neutrophil. So the neutrophil then detects or detects those smoke signals or those communication molecules produced by the macrophage while it's in the blood system. And it starts to says, okay, I know where the infection is. It climbs out of the blood vessel, and then it goes into the tissue to help that macrophage eliminate the bacteria. And neutrophils really have a lot of capability for eliminating foreign pathogens, and we'll talk about that in a little bit more detail as well. But one thing that you can think about, if you see data or if you see measurements of neutrophils in the blood or in the tissue, an increase in the number of neutrophils in the, let's say, in intestinal tissue or in circulation actually indicates that there's some kind of inflammation happening in the host or there's some infection in the host. So neutrophils are often used to determine kind of health status of the host. If they're high, then usually that indicates that there's some disruption in host homeostasis as an inflammatory response or some active infection going on in the host. So neutrophils are a nice cell to be able to measure to kind of give us an idea of what's happening in our host animals. So let's talk about phagocytosis a little bit more detail. So leukocyte phagocytosis, neutrophils perform this activity, macrophages perform this activity, as well as some other cells we're not going to focus on today. But what happens when the leukocyte detects that pathogen and phagocytoses it or brings it into the cell itself? And if you remember that picture of the macrophage under the microscope and you saw these appendages that were coming out of that cell, that's what these are right here that you see. Appendages come out and engulf the microbe or the foreign particle that the immune cell detects. So it brings it into the cell and it forms a membrane around that microorganism that becomes what's termed the phagosome. It then, inside the cytoplasm of the immune cell is a component called the lysosome. And the lysosome contains digestive enzymes. So the lysosome then merges with that phagosome and it releases those digestive enzymes so that the microbe is exposed to those digestive enzymes. It's chopped up, cut up, and killed. And that becomes the phagolysosome that we see here with the enzymes that now have digested or partially digested, broken up that microbe and killed it. And then this indigestible material is left and it really just gets expelled out of the cell now that the bacteria is dead and eliminated. So like I say, neutrophils do that. So that's how they go in, and they're very, very, very good at phagocytosing and killing off large numbers of bacteria with large numbers themselves. Macrophages also phagocytose, And we're going to talk a little bit more about the process when macrophages phagocytose. So they can kill the bacteria, like we saw in the previous slide, but they can also do something else called antigen presentation. And here's the schematic now. This immune cell is a macrophage. And here's the bacterium that the macrophage engulfs into the phagosome. It merges with a lysosome, and the enzymes chop that bacterium up into tiny pieces. But then the MHC class 2 within the cell takes a hold of one of those pieces of the chopped up bacteria and presents it on the cell surface. And what this really means, what it's doing is it's presenting it to the cells of the adaptive immune response. And this little cartoon here over to the right really gives a big picture of what's happening. The macrophage shown here as a little white monster-looking cell, and it's MHC2 in its hand with the bacteria particle on it. It shows it to the T cell, our little superhero T cell here, and it says, "Here, T cell, look what I found. Please help us eliminate this pathogenic material." So the T cell superhero of the adaptive immune response is specific for a certain bacterial component, and it says, "Ah, yes, I recognize it. Now I'm going to mount the immune response and help you eliminate this pathogen from the host system and bring back homeostasis." So antigen presentation is really key in a whole immune response because it links the innate and the adaptive immune response. And it really makes a whole orchestrated response focused on eliminating that pathogenic challenge in the host. So it's really key to that whole response and the subsequent things that follow. So let's talk a little bit more about that MHC, or major histocompatibility complex. And I don't know that this gets talked about a lot, but this major histocompatibility complex is the antigen presenting molecule on the cell surface of immune cells. And it actually has two types. There's an MHC class 1 and an MHC class 2. And the MHC class 1 is expressed on all nucleated cells. And so every cell within the host has MHC. If it has a nucleus, it has MHC class 1. And it really defines self. So it's unique to an individual, and it's how the immune system can tell the host that it's supposed to protect compared to foreign material like bacteria that it's supposed to eliminate. So this MHC class 1 molecule is called 1 because it has one segment that goes through the plasma membrane on the cell surface. In contrast to MHC class 2, which has two of those transmembrane segments that go through the membrane, and MHC class 2 is expressed on those specialized antigen presenting cells, like the macrophage that we just talked about in the previous slide. And its role really is being on the cell surface of those antigen presenting cells and presenting components of a pathogen to the adaptive immune response. And then when we talk about MHC, it's really interesting because, again, going back to that statement of MHC really defines or determines what the host, determines self. It determines for the immune system what is self or the host and what is foreign. And the MHC is associated with disease susceptibility in the host. So, and really more in the host species. So MHC diversity kind of explains some of this genetic link to disease susceptibility. So the MHC molecules are diverse within a species, but they're unique to, or mostly unique to, an individual. So that genetic link, so if you have a very inbred population, and the example of this, a great example of this is the cheetah. The cheetah population is very inbred. So they don't have a lot of genetic diversity within the cheetah species, and therefore their MHC diversity is low, meaning there are a lot of cheetah individuals that may have the same MHC as far as the genetic molecule component. And because of that limited genetic pool, cheetahs are very, very susceptible species to being wiped out by a single pathogen. So another way you can look at MHC is if you think about a flu virus that seems to impact a certain proportion of the population of the human species that may die off or be killed by a flu virus. Some may get very, very sick, but they don't succumb. And some they mount an immune response to that flu virus, but they really may not have any symptoms or know they were ever sick. COVID-19 is a great example of that. We know that there are certain populations of people that were exposed to the virus, may have mounted an immune response to the virus, but never had any symptoms of disease, while there are others that came down with the COVID-19 and succumbed to that infection. If the human population had a low genetic diversity and a low genetic diversity of MHC class 2, that virus could potentially wipe out the whole species. So in other words, MHC helps that genetic diversity of the species, the MHC class diversity of the species, have different susceptibilities or resistance to certain diseases. Now, how does that relate to our livestock and poultry? So there's lots of talk, particularly with our modern genetics in swine, where we selected very heavily for growth performance lines, specifically lean growth performance lines of pigs. And our modern pig really is genetically selected to most efficiently deposit feed resources as lean tissue. But with that genetic selection seems to have brought about a susceptibility to viral diseases. Viral diseases in our swine herds are a big problem. And a lot of the geneticists will say that our selection for lean genetics seems to have brought about inadvertently a more susceptible population to the viral challenges that are out there. And that's likely because the genetic diversity of the pig population decreased as we genetically selected individuals for lean growth, and therefore the MHC diversity decreased as well. So let's talk a little bit more detail about the differences between the two antigen presentation molecules, MHC class 1 and 2. We'll start with 2 because from an immune response, that one's really the one that is most integral for coordinating that innate and adaptive immune response to a pathogenic challenge. So again, we're back to the example of our macrophage with the microbe, breaks it up, puts it on its cell surface, presents it to the T helper cell. That T helper cell then produces cytokines or communication molecules like our smokestack, our smoke signals in the slide previously, and it communicates to other cells of the immune response like a B cell and a cytotoxic T cell. And it says to them with the smoke signals or the communication molecule cytokines, get ready, there's a pathogen out there and you're going to need to respond to it quickly. So now that essentially primes the B cell and the cytotoxic T cell to be ready to detect their antigens and then respond. In the case of MHC class 1 antigen presentation, and remember MHC class 1 is on the surface, the cell surface molecule on all nucleated host cells. So now in this target cell, it's been, let's say, invaded by a virus that has now incorporated itself into the cell. It's taken over the cellular machinery and is now generating peptide antigens that are actually viral components, not the host itself. So the MHC class 1 molecule presents these peptide antigens that are produced by the virus on its cell surface. The cytotoxic T cell says, I see that's a virus particle that's infected this cell. And the cytotoxic T cell then begins to eliminate that target cell. So it will essentially kill the target host cell by generating granular enzymes that poke holes in the cell surface and essentially eliminate that target cell that's virally infected so the virus can no longer use it to produce more viral particles to infect the host. So by eliminating that diseased or virus infected cell, it eliminates the ability of that virus to do more damage in the host. And then let's put that all together into one big picture for what happens when the immune system encounters antigen. So we've got the antigen shown up here in red. The macrophage here in the middle presents the components of the antigen to the T helper cells. The T helper cells then produce cytokines that communicate to the B cell that says get ready, but it also makes a memory copy of itself that's long-lived. So once the immune response is over, this memory cell is going to still be there. The B cell detects the antigen on its receptors on the cell surface. It makes plasma cells of itself that are the machinery to produce the antibodies. So these cells will do nothing but produce antibodies against that specific pathogen. The B cell also then makes memory copies of itself so that once this immune system's been mounted, it has more copies of these that can detect this specific antigen. And then if that antigen infiltrates or infects the host cell itself, such as the case of a virus, a viral antigen that infects the host tissue and the host cell itself, that infected cell displays the viral antigen on its cell surface. T cells get activated as cytotoxic T cells, and then they eliminate that infected cell that it detects the virus has invaded. And at the same time, it makes memory copies of itself. So then that whole immune system is coordinated really by the innate immune cells to the adaptive immune cells. And once the antigen is eliminated and the host is back to homeostasis, it now has memory copies of each one of these adaptive immune cells. So they're ready to mount that offense against the challenge much more quickly the next time it sees it. It's quicker because it can now skip the innate immune response and antigen presentation and immediately mount that immune response when it detects antigen again. So this kind of helps us start to understand this whole process for when we start to think about how do we interpret this immune data that we'll sometimes see in the real world. And really, the key for interpreting the immune data that we see in the real world is really kind of understanding this cytokine communication network. Most of what we measure in our animals or our host are going to be cytokines in circulation or cytokines that certain cells are producing. And we'll talk a little bit more about cytokines. They come in 6 different classes, and we're not going to go in just so you have kind of an idea of some of these terms. We're not going to go through each one of these in detail, but the chemokines here at the top are those molecules that the macrophage produce to call in the neutrophils, if you remember from the first part of the presentation. So chemokines, that's what they do. They're usually those communication molecules that say, hey, there's a infection here, and leaves kind of a trail for the other immune cells to follow to then get to the site of infection. And the other key group I wanted to point out are these tumor necrosis factors here at the bottom. And just because mainly one of the easiest cytokines to measure in a host is tumor necrosis factor alpha, which is generally associated with inflammation. So you'll hear these chemokines a lot, because they call in neutrophils, and you'll really hear this tumor necrosis factor, specifically tumor necrosis factor alpha, as an inflammatory cytokine that tends to be associated with reduced growth. Now, one of the things that I want to point out is the cytokines are numerous. I mean, this isn't even showing them all, but they're numerous. They can be termed either inflammatory or regulatory, and each cytokine can produce many that acts on itself or acts on other different immune cells within the host system. And a lot of them have what we call functional redundancies. So generally, tumor necrosis factor alpha is going to bring about an inflammatory response, but also there are other cytokines, interleukin-1, interleukin-6, interleukin-12, that also indicate inflammation. And some of these can have the same function. So you can see now when you start to try to interpret immune measurements for what do they really mean, the story is so complex that it becomes extremely difficult if you measure just a single cytokine to really see the big picture of what's happening in the immune response. So I've got this little square of art, a picture, and some of you art aficionados may know what this is, but many of you probably don't really. You may think this is a picture of a wave, or maybe just an abstract swirl. You really don't know from just seeing this small square of the art what the true big picture is. But when you start to look at the big picture, you realize that this is Van Gogh's Starry Night. And this is very similar to measuring only one aspect of the immune system. So if we only measure that one little square in the whole context of the entire immune system, it's really, really hard to see that big picture of how to interpret that one measurement. You really need to look at the immune system as a whole. Now, that becomes very complex when the immune system as a whole has multiple parts, multiple cytokines, multiple cells, cells that produce the same cytokines and communicate the same messages to different cells. What does that really all mean? We can't measure, I mean, we're lucky if we can get 10 to 20 aspects of the immune response measured in one experiment. And that really isn't even enough to only give us maybe a little bit more of a picture of that square of that whole picture. So when we're thinking about these immune measurements that we do, let's again bring our minds back to host homeostasis. And even bringing back host homeostasis as simple as balancing an inflammatory and a regulatory immune response. And I love this graphic that's shown here. It shows the kind of yin and the yang of an immune response. The yin being the regulatory cytokines and the regulatory cells, and the yang being those that are associated with inflammation. And what's really key here are these little arrows that are shown between the yin and the yang. Because if you have an inflammatory response, say with our tumor necrosis factor alpha cytokine here, the response to that inflammatory cytokine is going to be a regulatory cytokine, such as interleukin-10. So between the two, what you're looking at is an inflammatory response that then becomes regulated by the cytokine, the regulatory cytokine. And you don't want too much of either. Too much of either too much inflammation, you reduce feed intake, you reduce animal performance responses. Too much regulation, your animals become much too susceptible, say, to a pathogenic infection. What we consider immune suppression can come about from these regulations. So you need both. You need an inflammatory response and you need a regulatory response. So when you start looking for that balance within the immune measurements that we can measure, that helps to start paint the picture of whether or not there's an inflammatory response going on. Is there a regulatory response going on? And do you see both so that the animal seems to be in a host homeostasis? So then when you interpret the immune data in the context of that inflammatory response, you also have to consider host health as well as the host growth response, because that can give you a clue as to whether or not that response is in homeostasis or if there's a disruption in host homeostasis by disease. So now let's move on to some real data. So here's some immunology that we've measured kind of in the real world, so to speak, where we've looked at the effect of Salmonax, which is refined functional carbohydrates from a yeast cell wall. And we looked at it in the context of an LPS or lipopolysaccharide challenge. And LPS is a component of E. coli bacteria, and it's highly immunogenic, meaning the immune system kind of gears itself up in an inflammatory way in response to LPS. And if you look at this chart showing rectal temperatures or fever responses in these pigs, what you're seeing is this line in the purple here at the bottom is the PBS or the control. So the pigs were injected with the control saline solution. The pink line up here at the top shows the LPS response. So you can see when the pigs are injected with the LPS, 2 hours after that injection, they have a very robust fever response of 104.5 degrees Fahrenheit, which is a fever response for pigs. And then we administered the LPS challenge with a low and a high level of Salmonax. And what we really wanted to see was, can Salmonax bring about kind of a host homeostasis response in response to an inflammatory challenge? And that's exactly what it showed here. So if you really look at this 2 hours right after the LPS or LPS challenge or the control, you can see that LPS really boosted the fever response in these pigs, but Salmonax at either both the low or the high dropped that fever response down to much less than the LPS alone. And it actually seemed to resolve that fever response much more quickly in these animals, bringing them back to what's considered more of a normal host temperature. So Salmonax in this immune data showing body temperature fever response from rectal temperatures actually shows Salmonax regulates the pig's febrile response to LPS challenge to manage that host homeostasis. So another set of data that we've measured with the Salmonax product in pigs is looking at immunoglobulin concentration in the colostrum and the milk produced from sows fed Salmonax compared to sows fed the control. And you can see in this series of charts here, the purple being sows that were fed Salmonax. In each one of these, the immunoglobulin concentration, whether it's IgG in the colostrum, whether it's IgA in colostrum and milk, or IgM in colostrum and milk, the Salmonax fed sows always had a higher immunoglobulin concentration throughout colostrum, early lactation, and late lactation. So what this is really measuring is Salmonax's increase of those immunoglobulin concentrations in colostrum and milk are actually an indirect measure of that whole sow's immune response as well. So immune data from the sow and the sow's health is manifested by increasing her ability to increase the amount of immune components or immunoglobulins that she's transferring to her pigs through the milk. So this healthy sow, what does that mean in the context of her offspring? And these healthy sows that are fed Salmonax in gestation and lactation will actually improve birth weights in their offspring. So this schematic shows 8 different research trials that we conducted with Salmonax, again shown in the purple, compared to sows that were not fed Salmonax, and the magnitude of response in birth weight difference between the control and the treated sows. And what you can see here in almost all 8 of these studies, the pigs from the sows that were fed Salmonax had higher birth weights compared to the control sows. And this is an interesting kind of measurement here that I think probably deserves more attention for some of our sow longevity aspects than we think about. I just had the opportunity to hear a webinar put on by Dr. Billy Flowers at North Carolina State University. And he's really one of the most well-renowned reproductive physiologists for swine out there. And he gave this presentation where he made the point that optimizing birth weight in replacement gilts is critical for success in the breeding herd. And what he indicated was that if you were selecting gilts that were greater than 1.6 kilograms at birth, these gilts that were selected with the higher birth weights stayed in the herd longer and had more successful litters than their contemporaries with lower birth weights gilts. And what he meant by that is he said, even if those lower birth weight that are maybe less than that 1.6 kilograms, those lower birth weight gilts, even if they had higher body weights at weaning, still showed that they would not be successful as replacement gilts in the breeding herd. So really the key factor that he indicated when selecting your replacement gilts was to really look at birth weight, not necessarily the selection later on before they're going in to be developed into the gilt development unit, but actually go ahead and use birth weight as a selection criteria to eliminate some of those lower birth weight pigs. So I thought that was something that I hadn't heard before and thought that was really key. And all that really is, most likely, is the sow health transferring again, making that gilt at birth be much more developmentally advanced than, say, the lower birth weight contemporaries. So all back to sow health and how Salmonax can help improve that. Continuing to talk about healthier sows, also they have heavier weaning weights on their pigs when they're weaned. This study shows 9 different studies showing the effect of Salmonax on pig weaning weight. And of those 9, every single one, except for the single one, had an increase in weaning weight when the pigs came from sows that were fed Salmonex compared to the controls. And of these 9 studies, 5 of them were statistically significant with great magnitudes of weaning weight improvements with Salmonex fed to the sows. So again, that increase in the sow health increases the health and productivity of her offspring. Not only the health and productivity of her offspring, but the healthy sow, again, helped by Salmonex. When Salmonex was fed to these sows during gestation and lactation, the wean to estrus interval dropped by 1.5 days compared to the control pigs. So the control sows were at about 6 days in a wean to estrus interval, and the application of Salmonex to the gestation and lactation diets dropped that to about a 4.5 day wean to estrus interval, the time between when they were weaned and when they exhibited estrus for rebreeding. And then related to that, also the percent of sows bred in the first 7 days were increased when the sows were fed Salmonex. So overall, Salmonex, based on its improvement of sow health, can actually reduce the non-productive sow days by a day or greater in your sow herds. So then one more study to look at immunology in the real world and how it relates to animal productivity and performance. This study was conducted at Arkansas, and it was really to look at how does feeding Salmonex to the sows improve the subsequent performance or productivity of their offspring after they're weaned. So in this study, we compared these pigs that were from sows fed Salmonex to a group of pigs that were on a study that looked at zinc oxide and the improvement of zinc oxide when it's fed at a high level to these nursery pigs. So it's really kind of 2 studies, study A and study B. And study A was looking at the sows. So we had a group of control sows or a group of sows that were fed 200 grams per metric ton of Salmonex in the lactation ration only. And then in study B, that was going on concurrently, was a nursery pig study where we looked at nursery pigs fed the control diet or nursery pigs that were fed 2,500 parts per million of zinc in the nursery ration. And we took blood samples from each of these groups of animals to obtain data for immune gene expression analysis in these animals. And tumor necrosis factor alpha was measured as an indicator of an immune inflammatory response. So we were looking at how did these 2 aspects, Salmonex fed to the sow or zinc oxide fed to nursery pigs, impact the immunological status of the nursery pigs. And showing the data, we're going to start with the growth performance response. So if you look at this chart here, it shows the growth performance response of the nursery pigs that came from sows fed Salmonex or sows that were fed the control, and the growth performance of the nursery pigs that were fed either a control or a zinc oxide at 2,500 parts per million in that nursery diet. So to just reemphasize, the zinc was fed during the nursery, but this treatment response here was just the pigs from Salmonex fed to the sows. So both of these in the nursery were a basal diet, looking at the effect of Salmonex fed to the sow on the nursery pigs. If we look at the initial body weight in this nursery study, as typical that we see with the administration of Salmonex to the sows in lactation, we saw an increase in the starting body weight of those pigs that were put on trial that came from sows that were fed Salmonex compared to those pigs from sows that were fed the control diet in lactation. And then for the zinc study, that was all nursery pigs without any administration of treatments to sows. So they had the same body weight initially as they should have. And then if we look at the growth performance of body weight across the phases of this nursery period post-weaning, what you see is that both the pharmacological or high level of zinc oxide that was fed in the nursery, as well as the pigs that came from sows that were fed Salmonax in lactation, all had increased body weight relative to their contemporary controls. And this maintained all the way to the end of the nursery, where the pigs that were fed from sows fed Salmonax had a higher body weight at the end of nursery compared to pigs that came from sows fed the control diet. And same thing for those pigs that were fed high levels of zinc oxide in the nursery compared to the pigs that were fed a basal diet in the nursery. So when we look at the immune data in the next slide, what we need to be thinking about is how are we going to interpret now the immune data that came from these pigs in the context of this pig performance data. Both of the products in this study improved animal performance, but both of the products in this study had very, very different immune responses. So here we're looking at the gene expression of tumor necrosis factor alpha, the inflammatory cytokine. And what you see when we measured this from pigs in the nursery, from sows that were fed Salmonax in lactation, those pigs had a higher gene expression of tumor necrosis factor alpha, or a higher inflammatory type of an immune response compared to pigs that came from control sows. And if you look at the zinc oxide pigs, a different picture. Those pigs that were fed zinc oxide in the nursery had a reduction in the expression of tumor necrosis factor alpha, indicating that that inflammatory cytokine seems to be being regulated by the zinc treatment relative to their contemporary controls that were not fed the zinc oxide, which had a higher expression of tumor necrosis factor alpha, or in a more inflammatory state compared to those pigs fed zinc oxide. So we've got pigs from the sows fed Salmonax and pigs from the treatment of the 2,500 parts per million zinc oxide in the post-weaning diets that had very different immune profiles, but very similar growth performance responses in the nursery. So now we have how do we interpret an immune response of tumor necrosis factor alpha that was very, very different but had the same performance? And I think that we have to start thinking about, again, immunology in the real world is all about the homeostatic response of balancing regulation and inflammation. So in the case of that study, that increase in tumor necrosis factor alpha is an indicator of inflammation. And generally, an increase in inflammation or tumor necrosis factor alpha is generally associated with a reduction in growth performance response in our pigs and other livestock species as well. But this isn't always true, as our data just showed in this study previously. In that study, Salmonax fed to the sows improved growth performance of their pigs in the nursery, similar to the high levels of zinc that were fed in those nursery pigs. But those growth performance benefits from Salmonax fed to the sow and the benefits that came from feeding high levels of zinc oxide result from two very different immunological modes of action. One was inflammatory in the case of Salmonax, and the tumor necrosis factor alpha increase with Salmonax, and one was regulatory in the case of zinc oxide, where the tumor necrosis factor alpha measurement was decreased in those pigs that were fed the high levels of zinc oxide. So one thing that I want to rationalize how can two different immune responses both be a high level inflammation and a low level inflammation can both be perceived as good, we have to think of inflammation as critical to defend the host against pathogens and heal damaged tissue. So in that way, both inflammation as well as regulation of that inflammatory response are both important to maintain host homeostasis. And to talk about that in a little bit more detail, when we think about a pathogenic challenge, if we think about the refined functional carbohydrates of Salmonax, we know that the components of those refined functional carbohydrates can bind to E. coli and Salmonella cell surface receptors. And you can see examples of that in the charts here, where we have an agglutination assay that shows Salmonax improves or increases the agglutination of E. coli compared to a leading yeast brand and compared to the control. And then for salmonella, we have the graph on the bottom here that shows that Salmonax in the purple actually increased the inhibition of salmonella to adhere to the gut epithelium. So less salmonella actually adhered to the gut epithelium when Salmonax was in solution with it, relative to a competitor yeast product on the market. So the refined functional carbohydrates, we know can control the pathogens by adherence. And we know, based on the data that we showed, as well as the components of refined functional carbohydrates, that Salmonax can modulate immune responses really based on the beta-glucan component within the refined functional carbohydrates. Beta-glucans are a known stimulator of inflammatory immune responses. So Salmonax is bringing about an inflammatory response from its beta-glucans. And this inflammatory response, combined with the pathogen binding ability of Salmonax, can combine effectively to eliminate disease challenges and bring that host back to a steady state or homeostasis. And interesting now, one thing to think about though is certain measures of inflammation can actually appear to be reduced by Salmonax when Salmonax eliminates the pathogenic challenge. And you say, how can that be? That seems to contradict. But if you think, okay, by itself, Salmonax will elicit an inflammatory response. But let's say you have an E. coli challenge in your pigs, and that E. coli has LPS, and it brings up an inflammatory response to those pathogenic bacteria. If you put Salmonax in and it eliminates the E. coli, you now have eliminated the inflammatory response of E. coli that now appears to reduce the inflammation in the whole host system. So again, I'm going to bring back the message that both inflammation as well as regulation of that inflammatory response is important to maintain or bring about that steady state or host homeostasis. And to conclude with that, some concluding thoughts to this talk is for immunology in the real world and how we kind of think about immunology when we're trying to come up with solutions or understand our host health or pig health and pig productivity, is that there is no good or bad immune response. I often get the question, let's say I showed an increase in tumor necrosis factor alpha. The first question I'll get from the swine nutritionist is, well, is that good or bad? And I try to say, well, let's not judge the immune system response. Let's more try to understand that immune system response. Because we have to remember, again, going back to that schematic of yin and yang of immunology, the yin and yang of inflammation is really key to balance the inflammatory response. You also need regulation. And an inflammatory response can bring about a regulatory response that actually can be a very good response for your improving productivity. So again, try not to judge the immune response as good or bad. Try to understand the mode of action for what's happening there and how that brings the host back to homeostasis. Remember that different immune responses can result in very similar growth performance responses based on the data that I showed you today. So just measuring one aspect of that immune response, I often get the question, is there one aspect of the immune response that it can serve as a biomarker for pig health? And my answer to that is, there's certain ones that can help you understand it in context, but really no, there's no one measurement for a biomarker of pig health. And I think if you go back to our Starry Night picture by Van Gogh, and the one little square as that one aspect or one biomarker really doesn't tell you what that whole picture, big picture of the immune response looks like. And why is that? Well, the immune measurements, they're easily misinterpreted in isolation. So if you measure that one little square in the picture, you misinterpret really the whole picture of the art of the immune response. It's a dynamic system. It's always changing. It's always in flux. So there's a lot of different aspects of that are all changing simultaneously in that whole orchestrated immune response. And we're only measuring one point in time. We're probably only measuring a very few aspects of the immune response. And remember, there's lots of functional redundancies to those measurements. The tissue samples that you measure only give you one little small aspect of the whole host immune response. whether you're measuring that immune factor or factors within the intestine, within the liver, or within the blood, the immune responses are going to be very, very different. So tumor necrosis factor alpha may be high in the intestine, but at the same time low in blood. And how do you start to interpret that? You have to interpret it into the whole host homeostatic response or steady state. So again, to leave you, I want to just emphasize that immune data is complex, but it should be interpreted within the context of your herd health status and and performance response. If you measure an aspect or multiple aspects of the immune response and you don't know what your animal performance is or what your herd health status is, it becomes extremely difficult to understand what that immune factor means. And with that, I'll leave you with that as a last closing statement and be happy to entertain any questions you might have.